Hello and welcome to this Nature Research custom media broadcast titled Resistance to CAR-T Immunotherapy for Hematological Cancers. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Curiox Biosystems. In the last 5 years, the US FDA has approved 4 anti-CD19 chimeric antigen receptor T-cell products for relapsed or refractory B-cell lymphomas and leukemia. However, only around 30 to 40% of patients maintain a complete remission at long term. With us in the studio is Dr. Marco Ruella, the Scientific Director of the Lymphoma Program at the University of Pennsylvania. He'll begin the webcast by presenting research to improve CAR-T therapy for cancer by studying tumour evasion mechanisms and other limitations of CAR-T immunotherapy. Specifically, he'll describe uses of a combination of next-generation techniques to characterise mechanisms of resistance, perform preclinical studies, and develop new CAR-T products. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to Dr. Ruella.
Hello everybody. First of all, let me thank the organizers, Nature Research, and the sponsor, Curiox, for giving me the opportunity to present our latest data during this seminar. The title of the webinar is Resistance to CAR T-cell Immunotherapy for Hematological Malignancies. These are my disclosures. CAR T-cell immunotherapy has really been changing the way we treat patients with cancer, in particular patients with hematological malignancies such as B-cell lymphoma, B-cell leukemia, and multiple myeloma. And the key concept that is behind this breakthrough therapy is shown in the cartoon here to the left. Patient T cells are collected and they are edited using a lentiviral or retroviral vector that encodes the chimeric antigen receptor or CAR. This is a protein that, once expressed on the surface, is able to recognize a tumor antigen, for example, CD19, through its antibody domain, but it's also able to activate the T cell using the CD3 zeta and 4-1BB or CD28 co-stimulatory domains. Once the CAR T cells are engineered in the lab, they are infused back to the patient, where they will be able to recognize CD19 positive tumor cells, leading to the death of the tumor cell itself. So this is a cartoon. Of course, I wanted to show you how this CAR T cell work in real life, or at least if we look at the cells under the microscope. You can see here in green we have a leukemic cells. This is CD19 positive. And we have several CAR T cells that are mixed with the leukemic cells. And what you can see here, that when a CAR T cell recognizes the leukemic cells, it rapidly binds to it, causing the death of the leukemia cells. Once again, I can show you that the CAR T cell binds, and then rapidly the leukemic cells would explode. That's called apoptosis. So as you can imagine, this process happens many times in the patient. One CAR T cell can kill one tumor cells and move to the other, and so on. At the same time, they're also able to proliferate and persisting in long term. So thanks to this remarkable activity, the FDA has now approved several products for clinical use. The first one to be approved was developed at my institution, the University of Pennsylvania, and then licensed to Novartis. It's called tisagenlecleucel, and was the first one to be approved for any indication. And now several products, such as, for example, Axicell, Brexcell, Lysocell, and also two products for multiple myeloma, in particular Idacel and Ciltacel, are now available in the US for commercial use. So it's a very exciting time for CAR T-cell immunotherapy for hematological malignancies. So what are the results of these products if we look at the long-term anti-tumor effect? And I'm showing you here two examples of the longest follow-up available for one of the products. This is Axicell, which is a retrovirally transduced CAR-19, including a CD28 costimulatory domain. And you can see that in this form of lymphoma, that would have otherwise a very poor prognosis, we have about 32% at 5 years, as you can see here, and a little bit higher for follicular lymphoma, but in an earlier time point, 40 months, of patients that are without disease long term. So these patients, especially for the aggressive lymphoma, diffuse large B-cell lymphoma, can be probably considered cured. At the same time, we at Penn were testing Tisa-cell, which is a similar construct but includes lentiviral transduction in another type of costimulatory domain for 1BB. And as you can see here in our study, the results are very similar. We have here the follow-up at 5 years for both lymphomas, and you can see that about a third of patients, and for about 40% for follicular lymphoma, are without disease at long term. So from one side, I think it's very promising and exciting that a single infusion of engineered T cell can cause long-lasting remissions in patients that would have otherwise a very poor prognosis. But at the same time, we know that the significant portion, or actually the vast majority of patients in lymphoma, but also in leukemia, and likewise in multiple myeloma, will eventually fail CAR T-cell therapy. Together with the need to improve efficacy, these therapies are also associated with toxicities. And so I think it's important to understand what are the mechanisms of both resistance but also toxicity so that we can improve our approaches and make better CAR T cells for the next generation of patients. So we recently tried to summarize what are the mechanisms of dysfunction and failure of CAR T cell therapy. And so you can see here a lot of factors can affect the function of CAR T cell within the tumor microenvironment and the consequent failure of killing tumor cells. I always like to start with the so-called pre-infusion barriers. What do I mean by pre-infusion barriers? Well, I mean those cases where we are not even able to get to the point where we can infuse the CAR product. And we have, unfortunately, cases as such, for example, where the patient unfortunately progresses during manufacturing of the CAR that typically takes between 3 to 8 weeks. There are patients who unfortunately have manufacturing failure. There are patients that cannot access CAR T-cell therapy due to socioeconomic issues. And so it's important to consider also these factors a potential cause of failure after CAR T cells. But if we consider more the biological reasons for failure of CAR T cell, I included here three main categories. One is the so-called CAR T cell dysfunction. In other words, if we have a T cell that is exhausted or a T cell that has already gone through a series of proliferations and is not able to establish memory, well, then this T cell can fail to control tumors long term. On the other side, the tumor microenvironment is a very inhibitory site where CAR T cells find really tough to survive and to proliferate. And lastly, tumor cells themselves can develop strategies to avoid tumor killing. For example, probably one of the most studied one is the loss of the target, CD19 negativity, BCMA negativity, and so on. We did quite a bit of work on the role of apoptosis. But today, what I really would like to discuss with you is the role of the tumor microenvironment. So in this first part of the talk, we will be discussing the tumor microenvironment. And then in the second part of the talk, we will be discussing strategies to mitigate toxicity. So Punith Guruprasad in the lab has been running this project where the main question was, how does the tumor microenvironment inhibit effector T cells in the tumor microenvironment? And the key question that we wanted to address was what are the cell types within the tumor microenvironment and how do they interact with effector CD8 cells? In order to address this question, we use a model that we think is ideal to study the tumor microenvironment. And this is Hodgkin lymphoma. Indeed, in Hodgkin lymphoma, 99% of the cells present in the tumor are immune infiltrate and stromal cells. Only 1 to 2% of the cells are actually malignant cells. So it represents an ideal scenario to study, first, the tumor microenvironment itself, and two, understand what are the key ligands to receptor interaction that inhibit CD8 positive antitumor. And so to do that, we use single-cell RNA sequencing, and we analyze 4 Hodgkin lymphoma biopsies and use 2 healthy lymph nodes controls. We confirm this data in an additional 22 Hodgkin lymphoma biopsies. And as you can see here to the left, there are a lot of cells that are in the tumor microenvironment, and you can see in particular a lot of T cells, B cells, monocytes, and so on. And so we identify several groups, including, for example, the Tregs that are known to be inhibitory to the CD8 cells, and we ask the question, what are the key interactions between these two clusters? And we use the software called CellPhoneDB, which allows us to analyze single-cell RNA sequencing data. And the results were quite remarkable because, as you can see here, if we compare the interaction that are enriched in the Hodgkin lymphoma biopsy and compared to healthy lymph nodes, the BTLA-HVAM interaction is essentially the top enriched one in Hodgkin lymphoma as compared to healthy lymph nodes. So what is BTLA and what is HVAM? So BTLA is considered a checkpoint, immune checkpoint protein, which therefore is able to inhibit T cell receptor function through the phosphatases SHIP1 and SHIP2. In order to do this, it has to bind to HVAM, its natural receptor, which is either expressed on target cells, that could be tumor cells or stromal cells, but also can be expressed in cysts in the same T cell. Interestingly, it's a target that is starting to be explored also as an antibody therapy. You can see here 2 recent abstracts presented at ASH and ASCO of antibodies against BTLA. And so our hypothesis was, since we discovered that BTLA and its receptor HVAM and its ligand HVAM are essential in the tumor microenvironment to inhibit T cells, we hypothesized that we can knock out BTLA so that we can increase the function of CAR T cells against initially Hodgkin lymphoma. And in order to target Hodgkin lymphoma, we use CD30 targeting CAR T cell that we developed in collaboration with Apclone, which is a Korean company that develops antibodies and CAR T cells. And as you can see, we got a very nice expression of CAR30 in our CAR T cell. At the same time, the vast majority of the cells were knocked out for BTLA. So we were effectively able to get BTLA knockout, CAR30, using lentiviral transduction for the CAR and CRISPR-Cas9 knockout for BTLA. We initially tested CAR30, either BTLA knockout or wild type, in vitro against an Hodgkin lymphoma cell line. And as you can see here, the BTLA knockout CAR T cell were able to control the tumor much better than the CAR30 wild type. At the same time, not only they were able to control the tumor better, but they also had enhanced proliferation. So probably their enhanced survival and proliferation was leading to a better anti-tumor effect. And we confirm the enhanced proliferation here. You can see that clearly the cells in red are proliferating more as compared to the wild type CAR30 or the controls. Of course, we wanted to demonstrate these results also in a vivo setting, and so we use a immunodeficient mouse model, NSG mice, not SCIB gamma delta deficient mice, and engrafted them with an Hodgkin lymphoma cell line. It took quite a bit actually to get to see subcutaneous tumor, and we use a very low dose of CAR T cell, as you can see here, to treat our mice. And the results were very interesting because, as you can see here, the mice treated with control T cells, so these are T cells without the CAR, showed lack of tumor control. CAR30 wild type at this dose showed a benefit, but the benefit was minimal. It was only when we used BTLA knockout CAR30 that we did really see a benefit of our therapeutic agent. And this reflected into an improved overall survival, as you can see here, despite using very low doses, as you can see here, of CAR T cells. We next hypothesized that this could be a mechanism that could work not only in Hodgkin lymphoma, but for example, also in non-Hodgkin lymphoma. And so to do that, to test this hypothesis, we use CAR19 because non-Hodgkin lymphoma do express CD19, and we have several FDA-approved agents against CD19. And so we use a similar model, but using a non-Hodgkin lymphoma cell line, OCLY18, and we randomize the mice to receive 3 million CAR T cells at day 0. And as you can see here, again, the BTLA knockout CAR19 showed a much stronger anti-tumor effect as compared to the wild-type CAR19 or the controls. But of course, all these models are in immunodeficient mice, so there is no actual tumor microenvironment. And so, in order to study the tumor microenvironment, we switched to an immunocompetent murine model of lymphoma. We used A20 cell lines, which is a murine lymphoma. We used murine T cells that were knockout using CRISPR-Cas9 for BTLA. And we did do a lymphodepletion before infusion to ensure the functionality of our CAR T cell in an immunocompetent host. And again, the results were in line with our xenograft models. You can see here, again in red, the BTLA knockout mice were the only one that were able to control the tumor over time, while the wild-type CAR19 had some effect, but all the mice progressed, and the controlled untransduced T cell, they didn't lead to much anti-tumor effect. And of course, this reflected in a much longer overall survival for BTLA knockout, as you can see here. Lastly, in order to confirm that the mechanism was really mediated by the engaging of BTLA and the fact that in our most effective group there was no BTLA, we did the opposite. We tried to stimulate BTLA, and interestingly, the results was that when we stimulated BTLA, we actually got faster tumor progression as compared to control. So again, confirming that this pathway seems to be key in driving tumor responses to CAR T cells. So what is the mechanism? Well, again, we hypothesize that BTLA acts as an inhibitor of the T cell receptor and as a consequence also of the CAR receptor through the phosphatases SHP1 and SHP2. And so in order to test that there was less activity of SHP1 and SHP2 upon BTLA knockout, we perform phosphoflow cytometry. And as you can see here, the T cells that were BTLA knockout show reduced phosphorylation of SHIP1 as compared to the controls of CAR30. Lastly, in order to show that this effect is specific for HVAM, we tested a cell line that does not have the ligand of BTLA. And in this cell line, there is really no difference if you have or not have the knockout of BTLA. But instead, if you use the same cell line and you now overexpress HVAM, well, now you can see that there is a decrease of killing of CAR19, but BTLA knockout CAR, they can still kill very efficiently. Of course, there are a lot of strategies that are being developed to improve CAR T cell function. And so we picked the one that has been tested in the clinical and the one that has more clinical trial that are ongoing, which is PD-1 blockade. And so what we did in this experiment, and we go back to the CAR30 Hodgkin lymphoma model, we compared BTLA knockout and we added another arm with PD-1 knockout in yellow. And so you can see that the PD-1 knockout does improve a little bit the anti-tumor effect of CAR30, but not really to the effect of BTLA. And this is also confirmed in the overall survival, as you can see here to the right. Lastly, we wanted to get some clinically relevant data of the role of BTLA in CAR T cell therapy. And so we analyzed the CAR19 product from one of our trials. This is the trial for which I show you the 5 years follow-up. Is using CTL019, which then became the commercialized T cell. And we analyzed the product using bulk RNA sequencing. And interestingly, what we saw is that patients who had high levels of BTLA in the product, they also were the ones showing lack of response or partial response. And this was also true for the progression-free survival, where if you compare the patient with a high expression of BTLA and you compare to a low expression of BTLA, you can see that the high expressor have a very poor progression-free survival. We then moved to another CAR product, which is Axicell, and we reanalyzed the data that were published in Nature Medicine by the group of Good et al. In 2022. And what the authors did here, they did CyTOF analysis of the phenotype of CAR19 positive T cells that were sorted. And interestingly, BTLA was highly expressed in the group cluster 5 that was defined as CAR Tregs. And again, also in this setting, the patients with a high expression of BTLA tended to have a poor prognosis as compared to patients with lower BTLA. So we have described several mechanisms of resistance to CAR19, and I show you one specific project that we recently completed focused on the tumor microenvironment. And I want to spend the second part of my talk to discuss toxicities of CAR T cell therapy. And so, as any other very effective therapy, this therapy comes with very unique and severe toxicities. And in particular, I would say that cytokine release syndrome and neurological toxicity, also known as ICANS, are the two most common ones. So cytokine release syndrome is due to the fact that when the CAR T cell engage with the tumor cell, they secrete cytokines that in return activate monocytes, endothelial cells, and in general, stromal cells. They start to secrete IL-6 and other cytokines that increase exponentially in the serum of these patients. And as a consequence, these patients develop fever, they can develop hypotension, hypoxia, myalgias, and many other issues. And they can also develop neurological toxicity. They can go all the way from tremors to aphasia to and to coma. And so these are very serious toxicities that, again, are mostly due to the activation of the T cells and the consequent activation of the stromal and immune cells. Interestingly, when we think about CAR T cell therapy, we always think about the product itself. But there are many factors that can affect the function of these products in patients. And in particular, I think that we should include the lymphodepletion regimen. So this is the regimen that affects the function of CAR T cell and is the regimen that is given before CAR infusion. And as you can see in this table, we have several products against CD19 that are approved for clinical use. The first two, Axicell and Brexell, are characterized by CD28 co-stimulation, and the second two by 4-1BB co-stimulation. And you can see that these products lead to pretty different severe CRS grades. For example, you go 11% with Axicell, 15% with Brexell, and we go to 22 with Tizocell and only 2% with Lisocell. But the neurotoxicity data, I think, are particularly important when you consider the CD28 co-stimulated CAR have a very high rate of severe neurotoxicity, while the 4-1BB, they have less. But if we look at the lymphodepletion, the 4-1BB CARs, they have a slightly different lymphodepletion as compared to the CD28 co-stimulated. And in particular, there is a lower dose of fludarabine for Tizocell, and the presence also of bendamustine as a potential alternative. But cyclophosphamide is basically half of the dose for Tizocell, and also reduced in Lisocell, as you can see here, to 300 as compared to 500. So we hypothesize that the lymphodepletion regimens can actually affect the toxicity. And why would that be the case? Well, the first mechanism by which the lymphodepletion can affect T cell function, and the one that is the reason why it's given to patients, is that it prepares the cytokine environment for adoptive T cell therapy. In other words, increases the number, increases the concentrations of cytokines by reducing the lymphocytes that are the ones that would utilize those cytokines. But there are also the reason why lymphodepletion can be beneficial for T cell function and can modulate toxicity. Well, first of all, lymphodepletion can kill the tumor, but can also reduce immunosuppressive T regulatory cells that we mentioned earlier. And lastly, lymphodepletion can cause immunogenic cell death with antigen presentation, can lead to antigen presenting cell maturation, and also can damage the mucosa, the intestinal mucosa, leading to bacterial translocation that can stimulate the immune system. So in order to study the role of lymphodepletion during CAR T cell therapy, we look at the key lymphodepletion regimens that are currently used in the clinic. And the key one is fludarabine and cyclophosphamide. Cyclophosphamide is an alkylating agent, so it creates adducts in the DNA, blocking replication, while fludarabine is a purine analog, so it would intercalate in the DNA instead of regular purines, thereby blocking replication. Interestingly, another single drug called bendamustine is characterized by pretty similar characteristics, but takes a little bit of both drugs into the same molecule. It has an alkylating domain, but also a purine analog domain here in the middle. So bendamustine has been hypothesized to be a potentially beneficial lymphodepleting regimen by having characteristics of both cyclophosphamide and fludarabine, but is characterized in the clinic in non-CAR T-cell patients by a reduced toxicity as compared to both cyclophosphamide and fludarabine compounds. And so, in order to ask the question, does bendamustine lead to different toxicities as compared to fludarabine? Guido Ghilardi in the lab analyzed a large cohort of patients from Penn and from Portland and from Vienna, so a multi-center study including 132 patients with large B-cell lymphoma that were treated with the 4-1BB co-stimulated T-cell recursive. And the initial results were interesting because, as you can see here in red, it's fludarabine and cyclophosphamide, in blue, bendamustine. There is a clear increased effect of fludarabine and cyclophosphamide in reducing the number of lymphocytes after lymphodepletion. However, this very strong lymphodepleting action did not correlate with an increase on the overall response rate left, where you can see in both groups we have similar CRPR and progressive disease. And also, they didn't these two regimens didn't show any difference in the progression-free survival, as you can see here. But when we look at the toxicities, we were pretty surprised to see that bendamustine treated patient has a dramatically lower incidence of cytokine release syndrome, but also of neurotoxicity, as you can see here. And interestingly, together with these differences, we also saw that bendamustine was associated with reduced toxicity on neutrophils, so neutropenia, reduced decrease in platelets, so thrombocytopenia, and also higher levels of hemoglobin, so reduced anemia. And this is interesting because as a consequence, you have more infections in the fludarabine cyclophosphamide treated patients, and you also have higher levels of neutropenic fever, which is a very common infection in our patients. All in all, when we look at the hospitalization rates of these two lymphodepleting regimens, what we found is that in general, patients that were treated with bendamustine lymphodepletion were typically treated in the outpatient settings, much more than fludarabine and cyclophosphamide. And also that overall, if we look at the new hospitalization or in general, overall hospitalization rate, it was much higher in Cyflu as compared to Benda. After this study, there were still some open questions because we studied bendamustine in the setting of 4-1BB co-stimulating CAR T cells that are notoriously less toxic as compared to CD28 co-stimulated CAR. And also the question was, what is the mechanism by which bendamustine reduces toxicity? And this is particularly important given the toxicity of CD28 CAR, but also the fact that we have experienced on and off shortage of fludarabine over the last few years. And so we decided to analyze an additional cohort of patients that received axicabtagene autoleucels, and we analyzed patients treated with lymphodepletion with bendamustine versus fludarabine and cyclophosphamide. And as you can see here, the groups were well balanced in the two groups. There was a slightly increase of follicular lymphoma in the bendamustine, and this is justified because the approval for follicular lymphoma is more recent, and the use of bendamustine in this setting is also more recent. And so what are the initial data? Well, the overall response rate did not change. So very similar in the two groups as we saw for T-cell, and this was true in both diseases. Likewise, progression-free survival did not differ in the overall group, but also in the single groups. But again, when we analyzed the toxicities, now there was a significant reduction of cytokine release syndrome with bendamustine, and basically a 50% less neurotoxicity in the bendamustine as compared to fludarabine and cyclophosphamide. Importantly, also the infections were significantly different, together with the neutropenic fever that you can see is very rare with bendamustine. And as we saw for the T-cell gender occlusal, also in the CD28 co-stimulated CAR, the lowest value of neutrophil was higher for benda as compared to fludarabine and cyclophosphamide, like similarly to hemoglobin, and this was less marked for platelets. We then decided to look at long term. So we asked the question: is the difference at long term dependent on the lymphodepleting regimens? And at 6 months after CAR T-cell infusion, most patients showed like the median values for neutrophils, hemoglobin, and platelets were similar. But if you look at this distribution of the patients, you can see there are a lot of patients for fludarabine and cyclophosphamide that still have grade 1 or 2 um, thrombocytopenia, same patient that are anemic, and same patient with low neutrophils. So again, toxicity might be overcome at least for non-lymphocytes at 6 months, but still many patients are abnormal values. What's really remarkable, though, is that the lymphocytes were still different in 6 months, as you can see today. We then tried to start looking into the mechanism. And so the first question was, well, perhaps since fludarabine is giving a stronger lymphodepletion, well, then we have a stronger T cell activation and proliferation, and also a different phenotype of this activated CAR. And so we analyzed at day 7 the qPCR, and there was actually no difference. Like bendamustine and flu-cy patients were similar values of expansion. And then we switched to the analysis of the phenotype using flow cytometry. And to do that, we use the Auto 1000, which is a automatic cell processing system, including laminar wash. And why did we use that? Well, we used that because we wanted to make sure that the little material that we had from the peripheral blood of the patient was preserved. And so with the laminar wash, we were able to lose very little cells, certainly less than what we would have done with classic centrifuging and washing. But also doing the phenotyping, as you can see here, there was really no difference in a memory subset of these cells. So again, it was clearly not a direct effect on the. And so what was next? Well, we decided to look at pre and post lymphodepletion, cytokine expansion, and metabolomic factor. And so, very interestingly, we saw more cytokines being increased in fludarabine and cyclophosphamide as compared to bendamustine. And some of them are the expected one, for example, IL-15 and IL-7, and perhaps IL-2. But we also noticed several cytokines and chemokines like MAP, MIG, MCP1 that are typically, and also GM-CSF, that are typically associated with myeloid stimulation. And then perhaps this can be one of the clues connecting the increased neurotoxicity and in general toxicity in the fludarabine and cyclophosphamide as compared to bendamustine. We're now doing more studies where, for example, we're looking at the metabolism and metabolites in the serum of our patient treated with the different lymphodepletions. And again, very interesting results where you can see that there are strikingly different um, expression of these on these different metabolites in the two groups, with an enrichment in catabolism or and, um, reactive oxygen species in the in oxidative stress in general in the group treated with fluci. So in conclusion, I've shown you that for the first part of the project that the BTLA HVAM interaction is highly enriched in the Hodgkin lymphoma tumor microenvironment. And so we're now trying to understand if this is a generalizable path for other tumors. We did show that BTLA knockout enhances CAR T cell function. Now we're testing in more models. But importantly, BTLA seems to have a role in correlative studies of CAR19 from clinical studies. And of course, we're very interested in moving this approach of BTLA knockout forward to the clinic. In the second part of the story, we showed that Benda masking lymphodepletion has biological activity similar to fludarabine and cyclophosphamide, but is clearly characterized by reduced toxicities. And we think that higher levels of myeloid activating cytokines, in general inflammatory cytokines, might be causing this enhanced toxicity in fluci, because we don't really see a difference in number of CAR T cell in the peripheral blood. Interestingly, we did see a higher consumption of metabolites, so catabolism in fluci and antioxidant molecules, which potentially can be a cause of the metabolic stress. But of course, we need to do more studies to understand the actual mechanism. So I would like to thank all of you for your attention. And again, I would like to take the opportunity to thank Nature Research and Curiox for the invitation. I wouldn't be able to be here presenting without the work of my laboratory. You can see the people working in the lab listed to the left. In particular, I would like to thank Puneeth for the BTLA knockout project and Guido for the Mendamastin project. And again, this is a very talented group of people that I would like to thank. And again, thank you all of you for your attention. I'm happy to take any question if there is time.